Hello and welcome, everyone. We're going to start here in just a couple more minutes. And in the moment, as you are preparing for this, we welcome you to check the chat. On chat, you will find some information on how to best use this time, and also join us on our social media if you want to stay connected, and if you want to join our group that can help you find opportunities in this industry. All right, welcome everyone to this session. Today we're going to talk about our upcoming program called Bioinformatics for Precision Medicine. And before we jump into the program details, I want to introduce briefly who we are and what we do. So OmicsLogic is a training portal that connects biology and data science. And we are very focused on making sure that those who are learning about bioinformatics or data science and application to biological data are equipped with their necessary skills and experience that they can speak about what they can do, how they can apply it to different types of challenges, and how to use it. One of our major objectives with OmicsLogic is to address the question of how structured and how organized and systematized is this curriculum in the domain of bioinformatics. As you all know, there's a lot of free resources available on bioinformatics that you can find online. There are many free videos and courses and kind of different kinds of tutorials. And what we want to do is to really help biologists that already understand this domain, understand the kinds of questions that are being asked, get the most out of their training. Spend as little time as needed to acquire skills that they will actually use, and then develop a portfolio of projects that they can demonstrate in application to the challenges that they understand from a biological perspective. So as a result of working over the past several years, we have developed a series of baseline training in different omics data types, including introduction to bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, and a more advanced course on data science. And we kind of took that approach and applied it to several different domains, like oncology, infectious diseases, neuroscience, and then finally agriculture or biotechnology. Now, our approach is to make it accessible to people that have no background whatsoever in engineering, coding, or bioinformatics. And so we organize the coursework and skill acquisition according to the stage that you are at. So assuming that you have no previous experience, how do you actually start accumulating the kind of experience and skills that you need to really be successful in this field? And all of this is bundled together with different features that I will briefly describe in today's session. And then if you have any additional questions, I welcome you to actually try out this platform. And so before we go on, I welcome you to complete your profile. So all of you can, for free, register on the portal. It is learn.omicslogic.com. And you can join our growing community of professionals, students, and researchers from around the world. We have over 20,000 participants from top universities in the US and other places. And we welcome you to join them by trying out some of the features that we have built, and tell us what you like, what you don't like, and how we can really make it more useful. So our team behind this portal really is powered by both our vision, the founders that had the vision to develop this resource and to make it available, but also our team. And I want to mention a few people. Sparsh Dar is our marketing and sales team, leads our marketing and sales team, and she's going to be available if you want to contact her for any additional information about this program or other programs that we have going on. Her email is marketing@omicslogic.com. So the OmicsLogic portal really seeks to address three major goals. We want to provide training that enable independent research guided by mentors and peer example. We also want to build a community, and we have a growing community of over 20,000 participants. And we want to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. And one of the major applications of such skills and this domain in general is in precision medicine, which is what we will talk about today. So as we discuss the various topics and the associated resources that we have available to learn and master the skills in application to precision medicine, we'll really talk about how we can connect these molecular level details that are apparent in the genomic or omic data in general with clinical insights. How are they used? What kinds of achievements can we see that data has produced in these domains? And we'll touch briefly on several examples, primarily around the domain of oncology. So if you have joined us for the first time, we'd love to get to know you during today's session. Feel free to add your questions in the chat, and I will be addressing those as we go through the next few slides. Also, as I mentioned, if you have any questions at the end of this session, I'll be here to answer any of them during this webinar. And I hope that you can actually connect to some of the topics that we'll discuss and join us for the next few sessions that we have prepared around some of these topics. So the precision medicine program is really focused on 4 different sections. The first one is kind of at the level of understanding, what are clinical and molecular data that are used today in precision medicine? What does it mean? And how are they used, in different domains? Then we'll talk briefly about the types of molecular data that are most commonly used and how are they used to inform these kinds of questions and decisions that clinicians have to make. And that will include genomics, transcriptomics, and metagenomics. All 3 of these are widely used to understand, guide, and select the appropriate treatment. We'll also talk about how these kinds of insights are transitioning from the lab to the clinic. What are some challenges and opportunities, both in research, discovery, and then also in clinical use cases, and how are they different? And what are some of the challenges in research, and what are some of the challenges in actually application? And then we'll talk about some case studies. What are some data sets we've curated that you can start using to understand how this type of data could be used in genetic diseases or inherited, oncology, and infectious diseases? And that's not limited to this short list, but we have a library of resources that are curated according to different disciplines that you can explore. Let's start from the beginning. So first of all, how is molecular data informing clinical decisions, or how is it related to clinical data that is commonly being used by clinicians in the clinic? So the clinical data really describes disease symptoms, and the disease symptoms are kind of organized around major groups. So we call different diseases by different names. We can further kind of specify their stages or maybe their variation according to the patient's background. But the underlying pool of data that could inform the clinical diagnosis are these omics data sets that really tell us at a molecular subcellular level what is happening with this individual at this particular time, and what does that mean in terms of a very precise diagnosis, and how does that describe the biology that actually defines what that clinical phenotype is. So when a clinician looks at the clinical data, these additional omics data types have the potential to answer some of the more specific questions that they might have and help them make better, more precise, accurate decisions about what to do next. Let's take a look at how this plays out in such a field like oncology, where we have a growing burden of cancer around the world. So cancer incidence and mortality has been growing, particularly associated with older age, which is kind of characterizing this Western world. You can see US, Canada, and Australia kind of dominating how that population, which is increasingly older, and what is the incidence. So you can see that approximately 4 to 6% in these more advanced countries have in 2017 can have incidence of cancer. And it is the second dominant disease after cardiovascular diseases. And one of the reasons for that is because cancer is associated with cell division. And therefore, we kind of tend to see older people that have more cells that have been dividing for a long time. They tend to lead to cancer. And one of the major opportunities in terms of cancer is to try to identify the onset of cancer earlier on. Now, that poses a specific challenge, which is associated with screening an apparently healthy population. Previously, it was just cost prohibitive to be able to screen large sections of the population using omics data to try and identify these kinds of early onset risk markers. But increasingly, the technology that allows us to produce molecular data, which is next generation sequencing, is becoming more and more affordable, accessible, easier to use, and therefore widely used around the world, especially in these more advanced, economically advanced countries. And so we now see the availability of next generation sequencing as a technology that can help us understand what is happening at that cellular level. How are these cells dividing? How is this DNA being transcribed? And what are the abnormalities that start accumulating in different organs and tissues to try and detect early symptoms and early markers of increasing risk that these patients might have? Now, omics data in general, omics means collection. And so we can see omics being used for different types of data, really. And that can start from phenomics, where we try to characterize kind of visually how things look, tissues, cells, in a high throughput way. And then we can look at genomic data, where we sequence the genome or the whole exome, and we see specific mutations at individual nucleotide positions that we can try to associate with abnormalities. There's the whole field of epigenetics, where epigenomic data is being generated increasingly since about 2009, 2010, increasingly being informative because it really helps us understand the regulation that is not encoded in the genome that actually tends to result in the different levels of gene expression. And then finally, transcriptomics, which is where we can understand the actual expression of transcripts and genes, which of course lead to proteomics, which are the proteins being produced from these mRNA recipes. Now, in addition to these omics data, there's also viromics, metagenomics, and additional types of omics data that we can discuss, but these are the three most commonly used ones: genomics, epigenomics, and transcriptomics, and maybe today more and more metagenomics as well. So transcriptomics is one of those types of data that is really useful when we think about what makes a cell phenotype. So a cell phenotype, let's say we talk about tumor versus normal tissue, it is characterized by the proteins that are being produced inside that cell. And as you know, proteins are the result of the ribosome translating what is written on the mRNA, on the mature mRNA messenger RNA, into these structural functional proteins. And as a proxy, mRNA transcripts are a good way to evaluate how is that happening inside the cell, which allows us to really understand the presence, absence, and abundance of these recipes that are being produced. One example of how transcriptomics is being used is the ability to detect specific types of receptors like the HER2 receptor, which could be targeted by these monoclonal antibodies like trastuzumab. So really this opportunity to understand the transcription of different types of mRNAs in cells that become tumors or are tumors helps us understand how we can more accurately categorize many different types of cancers. And this, in fact, has happened already in the field of breast cancer classification. In fact, breast cancer classification has been developing from about 2010, 2013, where we see an introduction of additional subtypes that are actually categorized by these molecular profiling. And this molecular profiling is the next step after histological classification, where clinicians look at how different types of growths or tumors are contained within a specific tissue, or more invasive or infiltrating to other tissues. And they can also look at why that is happening. So look deeper at the molecular profiles and try to understand at the genomic, transcriptomic, proteomic, or even metabolomic level, how are these dynamics changing inside the cell? And a good way to kind of think about, well, how has that changed breast cancer classification is the appearance or emergence of luminal A, luminal B, triple negative cancers, for example, where we have molecular classification with specific kind of targets like HER2 protein, etc., and even functional classification to understand what do these cells have the potential to become. And so what that tells us about the role of bioinformatics that it plays in this field is that it really is a very specific approach that is data-driven to try to understand in the collected data that is available both in experimental data and clinical data of how we can connect the clinical manifestation of these phenotypes, like a disease such as cancer and its class and stage, with the underlying collection of different types of omics, and maybe even how they are related together. And the ultimate goal of this is to produce more precise and more personalized diagnostics and therapeutics. So let's take a look at some examples of how this has happened over time. So one of the interesting things about clinical use of bioinformatics and in general, this kind of approach to understand these molecular changes is that it really doesn't always make sense for us to start looking at the population already of clinical patients. A lot of times it starts with patient-derived cell lines, patient-derived xenograft models or animal models that have engrafted tumors in them, and then ultimately to try to use that information coming from cell lines and animal models to inform a better decision-making process when it comes to patients. And so what we will do in this program is take a look at some of these data sets that can characterize clinical populations using preclinical models, such as animal models, or even experimental data at the cell line level. So what ultimately this might lead to is how we can use molecular profiling to produce prognostic, diagnostic, and therapeutic markers, biomarkers like genes or mutations and other molecular changes to prescribe more precise clinical trials and ultimately treatment. Now, interestingly enough, over the past few years, starting from maybe 2015, we've seen an emergence of this new category of oncological drugs, therapeutics, that are called immunotherapy. That are actually today are becoming more and more effective and even surpassing chemotherapy, the traditional approach to treatment of cancer, by targeting specific receptors with monoclonal antibodies, oftentimes kind of triggering an immune response, so leveraging the immune response of a healthy individual. And one of those examples, as you can see here, Keytruda, has made a huge kind of change in this field in 2019. And then we can see here a specific targeted drug for the KRAS target. And just recently in June or July, July 2022, we saw this drug, dostarlimab, that demonstrated 100% complete response rate in a subset of patients with dMMR rectal cancer. Now, what's interesting here is that in this article, we read that these data provide the framework for immune ablative therapies and highlight the clinical impact of biomarker-driven therapy in early-stage diseases. Treatment of the tumor diagnostic dMMR population of early-stage diseases has the potential to eliminate the need for chemotherapy, radiation, and surgery in 3 to 4% of all cancers. This has the potential to be translated rapidly into areas around the world without access to modern chemotherapy, radiation, and therapy. And surgery, sorry. And so, as you can imagine, this has the potential to really change the way that we treat cancer, diagnose cancer, especially early on. And we read here about 3 to 4% of all cancers. This is an equivalent of about 28,000 people. So it's really a significant impact that will significantly reduce the cost of treatment, the ability to identify patients at risk, and to also improve their outcomes at the same time. Now, ultimately, the traditional approach is to try to kind of go from the clinical to the research, right? So a lot of times the traditional approach is that we take kind of the clinical phenotypes, the most impactful areas where a change could be made, and we start asking questions, what could be done differently? But in the modern era, there is an additional approach that is data-driven. And that is what I want to focus on in the next several slides of my presentation, is really talk about how availability of data that has been produced by top labs around the world that have generated sequencing data from cell lines, from animal models, and from patients can actually help us shorten the time from novel discoveries about patient subpopulations and subgroups, interaction between comorbidities or different types of diseases that kind of cause an ultimate decline in health, and the ability to prescribe new or existing treatments in the case of drug repurposing and repositioning. So where is this data? First of all, I'm sure that all of you are familiar with the National Center of Biotechnology Information, or NCBI, where over the past several decades there has been an exponential growth in the available data that characterizes different pathogens, individuals, animal models, experiments at the molecular level. And this really kind of brought this paradigm of big data into the field of clinical research and biomedical research. And so today we have a growing volume, variety, value, and velocity of data that is being generated. In fact, today we see that the pace of data generation is much faster than the ability of labs and groups that deal with this kind of research analyze data and extract meaningful insights. And what I mean by that is not the ability to produce reports, not clinical reporting on significant variants, but people who are trained to look at data, ask questions, and find indications in the data sets that could help us answer some of these questions that are so important and burning for patients that have only months to live. One of the important projects in this field, in the field of oncology, has been the TCGA, or the Cancer Genome Atlas. The Cancer Genome Atlas provides a unique opportunity to see multiple types of omics data combined together to describe approximately 13 different types of cancer. And this covers over 30,000 cases with data that is available for pretty much anyone to analyze. So here you can see an example of some of those clinical characterizations of patients. Of course, these patients are de-identified, so you have their ID only, but you can actually see different types of characteristics like comorbidities. In this case, this is a liver cancer population of liver cancer patients, but you can see hepatitis, age, BMI, obesity, and other factors. And interestingly enough, you also have a guide on how to effectively use such data and actually leverage some of the novel computational techniques to be able to extract meaningful insights from molecular and clinical data that is provided across these different types of cancer. Now, as I mentioned, there are many different types of cancer described or characterized in this project, but specifically when it comes to hepatocellular carcinoma or liver cancer, we can see that the variety of omics data, from exome sequencing to copy number variation to mRNA sequencing or transcriptomics to microRNA-seq, methylomics, and even proteomics, we can start finding significantly mutated genes and over-amplified genes, integrated subgroup classifiers, or how can we use these markers to identify specific patient populations, and additional insights that are critical to being able to treat one of these dominant cancers that are actually emerging in Asia and the Far East. So interestingly, there are both user-friendly tools and tools that are more kind of computational algorithmic to be able to identify these data. But I think the availability of this data has really brought about the awareness of how this publicly available data could be used both by clinicians and researchers, but also by citizen scientists that are able to find interesting insights and kind of develop further some ideas that could be proved based on real world evidence. Another important project in this field is the new kind of variation of genomic projects called the All of Us Research Hub, a new program by the National Institutes of Health here in the US, where we can see, again, different conditions across a variety of medical concepts, drug exposures, different types of medical characterizations and procedures. So a lot of clinical data coupled with genomic variants and additional type of data, including, by the way, transcriptomic and metagenomic data that is being generated. And all of this data is becoming more and more accessible, which is the really exciting part, I think, for those of us who are in this meeting. Now, in addition to that, we also have the NIH Human Microbiome Project, which again has over 30,000 individuals that have metagenomic 16S ribosomal RNA sequencing available for them, and allows us to understand the correlation between microbiome variation at the taxonomic level with different types of clinical and preclinical phenotypes. For example, exercise, age, diet, even ethnicity, and things like that. So it's really an exciting time to be in bioinformatics, whether you are a bioinformatician or clinician or a biologist, because ultimately availability of data is what has been preventing many from really applying their skills and understanding of data science methods and algorithms to extract interesting insights. For those of us that are not as familiar with what could be done, what we will do over the course of this program is use case study approach that helps us understand how experts in the field, from companies like Genentech, from research labs like MD Anderson, and even from clinical use cases, on how to use such data to address some of the pressing needs in the field of precision medicine, from oncology to neurosciences to other applications as well. Now, you might look at this and you might think that this sounds fairly complex. And I agree, bioinformatics is complex. Not only do you have to understand the terminology, you have to be able to apply technical engineering skills on data acquisition, processing, then analysis, visualization, etc., to be able to answer some of these questions. And for many biologists, that kind of looks like this, right? We're overwhelmed with new ideas like deep learning and different types of visual techniques that tend to show something that is only meaningful to engineers and not really to biologists. And so what I want to say is that this specific program is really not about that. I think we will have a glimpse of that for those of you who are interested, but primarily we want to make this as user-friendly as possible. And so for that purpose, I want to introduce you to one of the resources that you will have accessible during this program, which is the T-Bioinfo platform. Now, the platform really breaks down some of this algorithmic complexity and allows you to build visual interactive pipelines with every step of the way clearly explained with inputs and outputs that you will recognize from some of the publications that you probably read and understand, and has a direct link to the biology that you're interested in. Now, along the way, we will use some of the methods for analyzing, annotating, and interpreting the results of data analysis using a user-friendly interface and focusing on the relationship between the data and the interpretation of the analysis results, and ultimately introducing you to the potentials that some of these complex algorithms like data mining and classification can have for those of us who are interested to make an impact in these data-driven areas of precision medicine research and translational and clinical research. Now, over the past couple of years, at least, maybe 2 or 3 years now, everybody has been talking about infectious diseases and not anymore oncology on its own. So we will also take a look at some examples from infectious diseases, where we will take a look at some outbreak dynamics that could be characterized with omics data, again, genomics and transcriptomics. And we will take a look at several examples like the Ebola outbreak of 2015-2016, the current pandemic that is kind of still going on with COVID, but also some other types of applications of microbiome analysis that can help us understand the association between infectious diseases caused by viruses, bacteria, and other types of pathogens, and how the response of the immune response and the markers of the immune response can help us identify patients that are at most risk or the best targets for a specific type of intervention. Now, all of this is just to say that as you learn about the methods of data analysis, and the key here is the logic of data analysis, you will then be able to think how those types of analyses that you've learned and maybe applied to a specific case study could be applicable in the clinic as well. So let me tell you a few examples of how this could be used. Classification is a domain in machine learning where we have annotated data, which helps us identify, based on new data that we generate about a patient, a specific group that that patient can fall under. And some applications include clinical as well as pharma and biotech. So for clinical, this could be used for patient stratification, like grouping patients by risk levels, disease classification and diagnostics, identification of potential responders to therapies, and understanding of mechanisms of action or mechanisms of disease onset and progression. And then in pharma biotech, a lot of their focus is on discovery of biomarkers, right? So we just saw an example in an article how these kind of biomarker-guided treatment options are going to revolutionize the way immunotherapy is used and even eliminate the need for other types of treatments like chemotherapy and surgery. Also, detection of toxicity at the in vitro stage. So one of the drugs that we saw there, it is very promising and it has the potential to really affect this large sized population. But the problem is that it could have side effects and that it's not going to be useful for everyone. And so unfortunately, when there's a promise of efficacy, there's also a danger of toxicity. And if we could identify the toxicity in patients at the in vitro stage, that would ultimately lead to elimination of a lot of waste and actually an opportunity to focus energy and resources on more promising drugs. Analysis of molecular mechanism of a drug action or target discovery, a big field where we are seeing a lot of pharmaceutical companies kind of going through the FDA approval process and identifying that not only do we know that this drug works, but this is its biological mechanism of action. And increasingly, the mechanism of action is at the molecular level. And then finally, drug repurposing or repositioning. So all of these applications are applications of classification. And that is one of the methods that you can master very quickly by going through some of the case studies and examples that we will introduce throughout this program. Now, for those of you who are interested, these user-friendly tools are going to be coupled with scripting and coding, so you could learn how to implement some of those methods on your own. And the outcome here is to help you build out a portfolio that can illustrate how you can apply these skills effectively and build out your own set of projects. And that will be driven by tutorials and coding exercises that you can follow online on our portal. So to summarize, by joining this program, we hope that every participant will have the ability to understand scientific literature that describes analysis of omics data, will be able to perform basic processing steps for next generation sequencing data analysis, perform exploratory analysis and generate a hypothesis from data or data-driven research, design contrast for statistical tests and prepare data for machine learning, annotate found signal using biological databases and references, identify appropriate data sets for answering a biological question, and plan a bioinformatics analysis from start to finish independently. Now, for some of you, and maybe not all, I think an additional bonus is being able to present a project proposal, including the rationale, method, scientific background, and anticipated results. And if you are at that stage where you design a project proposal, we will then present you with additional opportunities to work with mentors that you could leverage to actually complete that analysis and leverage that project in your next research, clinical, biotech, or even innovative entrepreneurial application. Now, many students from around the world have completed such training. And importantly, if you come from a research background, you will be seeing how it's easy to take what you have learned and apply it to an area where you already are applying your research skills or participating in interesting projects. And this was the case with Olga Dartsova. She now works for a pharmaceutical company, but back then she was a postdoc at the Roswell Park Comprehensive Cancer Center and was able to apply the tools that she has learned to her own research as well. I want to conclude with a brief overview of program resources. What do you get by joining this particular program? You will have access to training materials that are both asynchronous and live, just like what we're doing now. As I mentioned, a lot of learning will be focused on the logic first, so you'll understand the connection between these important concepts and application of these concepts to real-world challenges. But if you are interested in, we will provide you with all of the source code as well and train you on how to develop a Jupyter notebook of your own data analysis process using R and Python. And then finally, all of the sessions will be recorded and available to you even after the program is over. Over the course of this program, we will be relying on the Bioinfo platform, which allows large-scale analysis, visualization, and integration of omics data. And this includes different sections for genomic data, like whole genome or whole exome data, transcriptomic data, epigenomic data, proteomic, metagenomic, and even biomic. And the goal is to allow you to quickly process, analyze, and visualize this data, and even generate interactive dashboards that you can then use to present what you have found. After testing this platform with many users, we find that the majority find it mostly or extremely useful. And importantly, it is built around technologies that are scalable and applicable beyond just the learning that you will see in this program. Researchers from top institutions have been using the platform to analyze their own data, and institutions like Oregon State University, the Mayo Clinic, or University of California, San Francisco. And as I mentioned, one of the key features is that you understand the logic of analysis and are able to apply this logic in a visual, reproducible way to extract meaningful insights. Now, these kinds of applications are important because it really simplifies and shortens the path to discovery and ranges from simple or fairly simple data sets to increasingly complex, including methods like single cell analysis, metagenomics and metatranscriptomics, genomic data analysis with characterization of individual mutations, and data mining and machine learning, which we already talked about being used increasingly in this field. Now, what we prefer to do is not just teach you kind of tutorial style where here's the problem, here's the code, here's what you need to do, but instead focus on case studies. And so we'll use these examples, extracting the abstract from publications that had an impact in this field and allowing you to understand how the terms explained in those articles are linked to actual data analysis techniques that you can both learn about and practice. And I want to mention that you can start with all of this today. So even if you are not able to participate in the program, we invite you today to join the portal. And so I briefly want to go to the portal and show you how you can register and sign up. So the portal link is learn.omicslogic.com. I will paste the link in the chat. Right here, you can register for free and create a free account. So you can go to login. You can use sign up right here. You can create an account right here on this portal. And I will be able to use my existing login to go into my profile. So once you have created your profile, you can then complete it by adding your profile picture, social media links, and a brief bio. And as you go through the different training parts in this program or asynchronously, you will see your progress being mentioned under the courses, your certificates being added to this tab right here. And today, you will be able to join the programs that we have. So the program, once you've signed up, the program is going to be available. I think, yep, so we have the link in the chat. This is the program itself. And you can see here how all of the information is available. Today we had the webinar, so you could learn more and share this with others in your network. And you can also start looking at the schedule that we have for this program. Once you go through the details and understand the schedule, you can see some of the resources that are available and select the level that you're more interested in, which is based on the time frame that you will have access to all of these resources, as well as some additional features like access to mentors and working on an independent research proposal, and actually completing your research project as well. And here you will see some of the case studies that will be available during this program. So at this point, I'm going to open up the questions. So if you have any questions, please do let us know in the chat. Let me stop